Welcome. My name is Armin Hadziahmetovic and I'm a doctoral student at the LMU Munich from Germany. I have the pleasure to present to you an outline on our method, EmpiReS, which can be used for the differential analysis of both gene expression and alternative splicing. The motivation behind this work is that the adequate handling of technical noise and biological variability is a challenging task. It has been approached by many different methods. Most often those methods employ some sort of model assumptions that may or may not hold true for a given data set. And therefore it is really important for the end user to understand the employed models and how they behave. Our goal with this poster is to show that we can reach a similar, if not even better performance in differential detection without relying on any model assumptions. Here we see a visualization of the difference between EmpiReS and a model-based approach. In our case, we rely only on the measured data by using the equivalence class counts and the derived empirical error distribution. So we have basically two tasks. First, we want to analyze the differential gene expression, for which it is enough to derive the differential error distribution. In the case of the differential alternative splicing, we also need to compute the doubly differential error distribution between the equivalence classes. Equivalence classes, they represent the smallest shared part of a set of transcripts which is consistent with a set of reads. They are constructed by assigning reads to its potential transcripts of origin. Here in figure A, we see how reads are assigned to their equivalence class. In B, we see the potential pairs that are then later evaluated for each transcript pair in order to determine if they are differentially alternatively spliced. For the empirical error distributions, the log2 error between the replicates is computed and divided into varying intensity bins. In order to compute the differential error distribution, the two error distributions of the respective conditions are subtracted, and this is performed for each equivalence class. In order to obtain the doubly differential empirical error distribution, the differential error distributions of the different equivalence classes are subtracted from each other. And the observed change of changes of the pair of equivalence classes and the pair of replicates can be transformed into a z-score using a modified Stauffer's method. And now, in order to evaluate the performance, more specifically the sensitivity and specificity of the methods, we mainly relied on simulations based on real data. So the simulation of differential events is performed by swapping counts of observed measures in one condition while the other remains unchanged, and so introducing artificial differential events. This of course requires that we have a big replicate pool of the same condition that we can divide then into two conditions which per definition should not exhibit any significant changes between them. So this is really important here. In summary, this means we have no counts which are simulated, but we instead only take actual observed counts which best fit our need for a specific fold change. Shown in the figure here are the F1 scores of the different methods across 6 of the data sets. And we see a clear difference between the data sets. So for DEXIC, for example, we see that 2 of the data sets match or perform really good while 4 of the others do not match or apparently do not match the model assumptions. In the second figure, we see the performance of the transcript-centric methods on the same data. And here we also see that we have some varying differences, and the overall takeaway point here is that EmpiReS performs best, or at least is most robust in respect to the different datasets. So, in conclusion, we have shown that we do not have to explicitly model the measurements. We have seen that certain model assumptions work good for specific datasets, but may also perform much worse on other datasets. And we have also shown that our approach works well for doubly differential comparisons, such as is the case for differential alternative splicing. And with that, I'm concluding this brief poster presentation, and I want to thank you very much for listening.